pollen grains have been preserved in liquid nitrogen for several years, and have a temperature of -196 degrees Celsius. Cryopreservation is the name of this form of saving. The low temperature allows for a longer period of storage, as it decreases the rate of cell growth. Each mother cell on reductional division produces 4 microspore tetrads, also known as pollen. So, 4 microspore cells are formed from 1 pollen mother cell. So, 1 microspore cell are formed from 1 by 4 pollen mother cells. So, 64 microspore cells are formed from 64 into 1 by 4 equals 16. Therefore 16 reductional division are required to form 64 mother cells.
Endosperm is triploid in nature. That is 3n. So, 3n equals 24. Then, n equals 24 by 3 equals 8. Each cell of the root structure represents the diploid condition in the plants. That is 2n. So, 2n equals 2 times 8 equals 16.
At maturity, the gametophyte is 7-celled and 8-nucleated.
Endosperm is triploid in nature. That is 3n. So, 3n equals 24. Each cell of the gamete represents the haploid condition in the plants. That is n. So, n equals 24 by 3 equals 8.
A megaspore mother cell, or megasporocyte, is a diploid cell in plants, in which meiosis will occur, resulting in the production of 4 megaspores, generally, within each sporangium. Meiosis of a megaspore mother cell produced 4 haploid megaspores. In gymnosperms and angiosperms, only one of these 4 megaspores is functional at maturity, and the other 3 degenerate. So, one megaspore mother cell, upon meiosis, gives 1 viable megaspore. Typical angiosperm anther is bilobed, with each lobe having 2 theca each. So, the angiosperm anther is dathecous. Hence an anther has 2 lobes with 4 theca.